Hola. Unfortunately, that's the end of my Spanish. I'm really embarrassed to say. I took 2 years in high school, but that was a very long time ago. So thank you to Janine in the back, who will hopefully help me communicate with you through these devices. My name is Michael Ostland. I'm with Denali Therapeutics. I want to say a big thank you to the organizers and to all of you for giving me the opportunity to talk to you today about our company, a few programs that we have in MPS and specifically in Hunter syndrome. I'm really happy to be on this panel with other industry colleagues. I think we all know in this room what a difficult disease this is and how important it is to have many opportunities to bring therapies to patients. And all these efforts take a great deal of collaboration between us and industry, expert physicians and investigators, regulators, and of course you, the families. So we're very happy to be here today. A few disclosures. I am an employee of Denali Therapeutics. I will be talking about a few molecules that we have at Denali. None of those molecules are approved by any health authorities. They are investigational drugs at this moment. So I'd like to just spend a minute introducing you to Denali. You may not know much about Denali. We're a fairly young company. It was founded in, and I should check this out. Yes, very good. We were founded in 2015 with a purpose to defeat neurodegeneration. We are based in San Francisco, California, but we also have facilities we have capabilities globally and an office in Zurich, Switzerland, which was founded about a year ago. That's my job. I lead that office. And the goal there is to really bring us closer to the European community to enable us to run trials and work closely with stakeholders here in Europe. We have 7 programs that are currently in clinical studies, and we are firmly committed to collaboration as a means to success. Our disease areas of focus, again, it's all neurodegeneration, but that includes Alzheimer's, Parkinson's, ALS, and the topic for today, liposomal storage disorders like MPS. Denali is very committed to science, and we have 3 core principles that we use across our entire pipeline. I think all 3 of those principles are evident in our programs in MPS. And I think this audience understands very well the genetic pathway relevance to MPS. We also work to engineer molecules to cross the blood-brain barrier, and I'll say a bit about that on the next slide. And everything we do relies heavily on using biomarkers, so measurements in the patient that help us determine proof of concept and also understand the appropriate dose for our medicines. So I'd like to spend just a minute talking about the enzyme transport vehicle, or ETV technology at Denali. And I'm sure this audience, we all know, or you know better than I, that the neurologic manifestations of MPS are really devastating and require treatments that can get into the brain to address these. And this is a core problem in drug development. Our ETV platform is our strategy in order to get enzymes into the brain. I think you'll hear from this panel today that there are multiple strategies for this, and this is a good thing. I know that football is an important thing here in Barcelona, so I'll use a metaphor that we need lots of shots on goal. And you need diversity of approaches in order to score those goals. So I think it's a really great thing that the science that is breaking, that we heard about before the coffee break, allows a diversity of approach. At Denali, we're very excited about our approach, and we think it has a real promise. But ultimately, we're humble enough to know that we need to prove this in clinical trials. And that's what I think we're all trying to do together in this community to figure out what works well. So just to sketch the ideas behind the enzyme transport vehicle, this diagram over here, let me try this. Does this work? Yes, it does. So this diagram right here depicts the blood vessels here on the left, where it's easy to get medicines or treatments into, the brain, where we are attempting to get medicines, and this so-called blood-brain barrier that is a barrier for getting larger molecules across. And the idea that we're using, which was discovered by some brilliant scientists years ago, was to take advantage of a normal process that is used to transport iron across this barrier. So a part of the body uses this transferrin receptor, TFR, to transport iron across the blood-brain barrier. So the idea is that we want to hitch a ride on that same transporter and get therapeutic enzymes across. The graphic over here shows you a depiction of the enzyme here in the blue circle, and then the innovation for Denali is a very specific linker that we manufacture attached to the enzyme, but that will also bind attached to the TFR receptor. And our strategy, and we work very hard, is to come up with a binding that is sufficiently tight, but not too tight. So tight enough to latch on, but also loose enough that when it gets across that it will let go. So this is the strategy, and this is what we hope to see in our upcoming clinical trials, that this translates into benefit for patients. Just a few more words on our overall pipeline at Denali with respect to MPS. I'm here to talk to you mostly today about our DNL310 program in MPS II, which is in late-stage clinical trials. But we also have programs that are a little bit further back for Sanfilippo, MPS III, and also MPS I, Hurlers. And that's why it's a real pleasure for me to be here today, because I think this will be hopefully a partnership that goes on for many molecules for much time. So now I'd like to transition to speak to you about the specific program, DNL310 in Hunter syndrome. And this graphic is just maybe recapping a few things that I said earlier. The DNL310 is an example of the ETV platform that I described to you that's delivering the IDES enzyme through the brain and the body. So it's delivered IV, and then using the transferrinase receptor that we discussed, we expect broad distribution into the brain. And there's also a potential that using this, the TFR, that it can distribute enzyme into tissues such as bone, cartilage, and the heart. So that's something we're excited about, that potential, and we look forward to seeing what we learn in clinical trials. So altogether, DNL310 has the potential to treat both the neurodevelopmental and physical symptoms of MPS II. So I have one slide to give you an overview of our clinical studies in Hunter syndrome with DNL310. The top bar here is our ongoing phase 1/2 study in MPS II. The objectives of this trial were to assess safety and also the pharmacokinetics and pharmacodynamics. So how much drug is getting into the body and what it's doing. About 5 weeks ago in Germany, Dr. Munzer presented some interim data from this ongoing trial, and we now have 27 patients with up to about 18 months of follow-up. And these are the headlines from that presentation. That I mentioned the importance of biomarkers earlier for guiding us. We see that DNL310 is able to rapidly normalize the CSF heparan sulfate, which, as you know, is a really key gag for understanding this disease. it's not just that we're reducing it, but we're actually bringing it into levels that we see in unaffected patients. We're very excited about that response in this small trial. The safety data, very important. We studied multiple doses in this trial, and broadly speaking, the safety were consistent with enzyme replacement therapy. We have an independent data monitoring committee that's closely reviewing these data. There's been no discontinuations due to adverse events or withdrawals. So a manageable profile that allows us to continue at all the doses that we studied. So based on that safety profile and the biomarker evidence, we were able to pick a dose for our phase 3 trial and start initiating this phase 2/3 clinical trial, which is on the bottom of my slide here. This trial has a fancy name, COMPASS, and the objective for this trial is to determine the efficacy and safety so that we can hopefully, if positive, bring this entire data package to health authorities for approval. That is our hope. All the information on these trials is, of course, available on clinicaltrials.gov. So just a quick overview of the COMPASS study. So this slide is giving you in words on the left and in pictures on the right, so you can pick what side you like. But broadly speaking, there are 2 cohorts that we are studying in this trial. Cohort A, which will be in neuronopathic MPS patients in children aged 2 to 6 years old. Cohort B, this is the non-neuronopathic cohort, and these are children aged 6 children and teens, 6 to 17 years. There are other inclusion criteria, of course, and a complete list can be found on clinicaltrials.gov. In each cohort, we will randomize patients 2-to-1 to either DNL310, the investigational agent, or standard of care IDS enzyme replacement therapy. So for every 3 patients, 2 will be on active, 1 will be on the standard of care, and it'll be blinded. Neither the patients, families, physicians, or raters will know who's on which treatment. The plan is to enroll 54 subjects globally in North America, Europe, Latin America, and Australia. The trial is open in North America. We dosed our first patient 3 weeks ago. We're very excited about that. In Europe, we're working very hard, and specifically, we expect 3 sites in Spain, in Barcelona, Madrid, and Santiago to open in the next few weeks. And it's part of my job and the Zurich team to help support that. The study length is 2 years in the neuronopathic cohort and 1 year in the non-neuronopathic cohort. After the trial completion of that primary period, all participants will have the option to enter the open label extension and receive DNL310. Just a few frequently asked questions. I think I already touched on this question of how it is randomized with the 2-to-1 randomization. That participation and eligibility is, of course, a more complex process than the high-level summary I've given you here, and it will be determined by doctors at each clinical study location. And also the double blinding, which I mentioned. This is a very important feature for us in that it will ensure that the integrity of the endpoints that we're assessing, because these are neurologic endpoints such as behavior. So ensuring that no one knows what treatment people are on will help the integrity, help these results be greeted with the highest integrity by health authorities. So with that, I want to say a big thank you, muchas gracias. I'll close with the other Spanish that I remember. And to you, the family and children participating in our trials and hopefully our future trials, the MPS Lysosomal Association here in Spain, and also the physicians and study teams that we're collaborating with on these clinical trials. So thank you very much.